Hi, welcome back to Biology classes with Lakshmi Chandra. In the previous sessions, we were dealing with the chapter Principles of Inheritance and Variation, wherein we had learned the various principles put forward by Mendel, some of the aspects which deviated from Mendelian principles. We learned about genetic disorders. We learned about their causes, their effects, and so forth. In this session, in this present session, after that we have learned the principles of inheritance, we will now see what is the molecular basis of inheritance. So we are dealing with the molecular basis of inheritance in this video. When you say the molecular basis of inheritance, all our mind goes towards that particular molecule which is responsible for inheritance. And I hope you have guessed it right. It is the DNA molecule or the deoxyribonucleic acid. So, it is very important for us to know regarding the structure of DNA so that we understand what is the molecular basis of inheritance very clearly. So, this video, today's session is devoted towards that, regarding what is the structure of DNA, what it is made up of, how are the different components in the DNA bonded, and why then this DNA has become the genetic material. So let's focus on that particular aspect. So we are moving on to the structure of DNA. So as you know, DNA is deoxyribonucleic acid. Deoxyribonucleic acid. This is our DNA. Deoxyribonucleic acid. When you learn the structure of deoxyribonucleic acid, first we will concentrate on about this particular aspect. What is a nucleic acid? What is the general structure of a nucleic acid? And then we shall relate it to our DNA, deoxyribonucleic acid. So let's first concentrate on what is a nucleic acid. Nucleic acid is made up of many nucleotides. They are made up of many nucleotides. So I can write like this: n number of nucleotides, or a polymer of nucleotide is nucleic acid. Like we say, a polymer of amino acids. When amino acids are linked one by one, one after the other, you are getting a big molecule, a macromolecule called as protein molecule. Similarly, when nucleotides are linked one after the other, you are getting a macromolecule called as nucleic acid. Now let's see what this nucleotide is made up of. Nucleotide is made up of two components. They are the nucleosides and phosphate group. Nucleoside and phosphate. So nucleotide is made up of nucleoside and phosphate. Nucleoside and phosphate. We all know regarding phosphate, isn't it? We all know regarding phosphate. If I write it generally, the structure, valency of phosphorus is 5. So I am putting here like this, phosphate linked by double bond and then 3 single bonds. These single bonds are all connected to OH. So this is our normal phosphoric acid, or if it is in the aqueous medium in the ion form, you call it as phosphate. So that is with regard to phosphate. Phosphate everyone knows, it is very clear. Now our focus is what is this nucleoside? Nucleoside. Again, nucleoside is a combination of again two other molecules. Which are they? It is made up of a pentose sugar, and a nitrogenous base. Pentose sugar and a nitrogenous base. Now what is this pentose sugar? What is nitrogenous base? Everything will be dealt with one after the other in this video. Okay. So from here we understood that nucleic acid is made up of nucleotides. Nucleotides are made up of nucleosides and phosphate, and nucleoside is made up of pentose, sugar and nitrogenous base. So I can say in general that nucleic acid is made up of the first component pentose sugar, the second component nitrogenous base, and the third component phosphate. So in general I can say a nucleic acid is made up of pentose sugar, nitrogenous base and phosphate. Next, let's see what is a pentose sugar and what are the nitrogenous bases one by one. So first I'll tell you what is a pentose sugar. A pentose sugar. I think you all have heard about glucose, fructose, galactose, mannose, isn't it? It's all ending with O's likewise. So they are all sugar molecules, simple sugar molecules. First let me take you the example of glucose. You are all familiar with glucose. How do you write structure of formula of glucose? You write it as C6H12O6. Often when we write the equation for photosynthesis and all, we use this particular formula, isn't it? C6H12O6. It is pointing towards a glucose molecule. So here you see, look at the number of carbon atoms. There are 6 carbon atoms here. So I am putting this glucose under the category of hexose. Hexose is a sugar having 6 carbon atoms. So I can say glucose is an example of a hexose. So hexose is a sugar having 6 carbon atoms. Fructose is another example. That fructose is also having 6 carbon atoms. But the arrangement of H and OH molecules are different. But still that is an hexose. So glucose is a hexose molecule. Fructose is hexose. Why I am calling that as hexose? It is all because it is having 6 carbon atoms. Now you can guess what is the meaning of pentose sugar. Isn't it? I told you hexose is having 6 carbon atoms. So naturally pentose is a sugar molecule having 5 carbon atoms. So pentose sugar has got 5 carbon atoms. Which is the example? The example of such a sugar is ribose. Ribose is example of a pentose. Like glucose is example of a hexose. So our point of attention is towards this one, ribose. Now you can just connect it here. See, deoxyribonucleic acid. Something related to that ribose is coming here. Okay, so let's focus on that one. Ribose. First, I'll just draw the structure of ribose. This is a simple way to draw the structure of ribose. I'll just tell you children, I'll just connect it with another molecule. When you draw a structure like this, we all know this is pointing towards a 6 carbon compound called as benzene. It is a cyclic compound, benzene. So here in that ring, I haven't mentioned any molecule's name. I haven't put any C, N, H, nothing. But it is understood that in these areas, the molecule present in these areas, it is the carbon. So in a cyclic compound, if you don't write anything at the corners there, it is understood that it is made up of carbon. Carbon is present there. But here you see here in the ribose, I have put here one circle. That circle represents oxygen. So in that area, you have oxygen. But in other areas, you are having carbon atoms. Okay, so let me just put the number for this carbon atoms, because I have told you there are 5 carbon atoms. Let us see whether 5 carbon atoms are there or not. So first carbon is here, this position. Second carbon is here in this position. third carbon here and fourth carbon here. Oh, Miss has told that there are 5 carbon atoms, but here there are only 4 carbon atoms. Where is the 5th carbon atom? In the case of ribose, the 5th carbon atom lies outside this particular ring. Okay, the 5th carbon atom lies outside the ring. It is here. It lies outside the ring. Okay, and here it is bonded to CH2OH. This is the structure. CH2OH. So in the rest of the areas you have carbon atoms, and in order to satisfy the valency, we know the valency of carbon is 4. In order to satisfy that valency, we have to put here other molecules. So the other molecules put here are H here, OH here. The second carbon also H, OH. The third carbon again H, OH. The fourth carbon, it is related to CH2OH and then another H. This is the structure of ribose. Structure of ribose. Okay. Now, I'll take your attention towards another aspect of this particular structure. You look at the arrangement of H and OH here. In the first carbon alone, concentrate in this part. In the first carbon, H is above and OH is below. Isn't it? This kind of ribose, you call it as the alpha ribose. In alpha ribose, what is the difference? What is the structure? How do you understand? Let's just look at the first carbon. If H is above and OH is below, you call it as alpha ribose. How to make it beta ribose? How to make it beta ribose? Let us see. How to make it beta ribose. Again, the same structure I am drawing. Okay, same thing I am drawing here. In the first carbon, just invert the position. OH above and H below. Rest of the areas there is no change. Rest of the areas there is no change. CH2OH and H here. But only in the first carbon I changed the position of H and OH. So this what I have drawn now is beta ribose. In our DNA, we are having this beta form. This form exists. But then again another question is here. Our DNA is not actually ribose. What is it? It is deoxyribose. It is deoxyribose. So what is the difference between ribose and deoxyribose? That is the next question. Okay, next question is what is the difference between ribose and deoxyribose? From the word itself it is clear. It is deoxy. Deoxy means one oxygen. Oxygen is less. One oxygen molecule is less, or it is in lack of one oxygen molecule. One oxygen molecule is becoming less somewhere from this ribose, so that now the ribose is getting converted to deoxyribose. So that is a question. In which position that oxygen molecule is less? Now it is with regard to the second carbon here. Here I have put here H and OH, isn't it? Instead of OH, if I am putting it as H and H, H is here the same. Instead of OH, that O I have removed and I have put there H. So one oxygen I have removed. From which position? From the second position. That position is very important. You all must keep this in mind. These things are asked for competitive examinations. Okay, so in deoxyribose, what is happening? In the second position of that ribose, one oxygen is removed. It is less there. So now the ribose has become deoxyribose. So in our DNA, this deoxy, so now I cannot write here ribose, but I am striking this and I am going to write here it is beta deoxyribose. It is beta deoxyribose. It is not ribose now. So in our DNA, this is the pentose sugar present. This is the pentose sugar present. Which is the pentose sugar? Deoxyribose. Now again I will write the structure and show to you how to draw this deoxyribose sugar. Very simple. The first carbon, it is a beta form, so it is HOHH. The second carbon, there is one oxygen less, there is no oxygen say there, so it is HOH. In the third carbon it is HOH here, CH2OH, H. So this is the area we have to concentrate on, this, and here it should be beta. So this is the structure of deoxyribose, beta deoxyribose. Now let us look into the next component. So pentose sugar is over. The next component was nitrogenous bases. Let us look into that one by one. Moving on to the nitrogenous bases. Nitrogenous bases. Nitrogenous bases can be categorized into two. They are the pyrimidines, and the purines. The pyrimidines and the purines. Two nitrogenous bases. Pyrimidines and purines. Okay, pyrimidines and purines. So what is this nitrogenous base actually? It is a heterocyclic compound. Okay, as I have shown you in the last, in the previous area that when we were dealing with ribose sugar structure, there I had drawn the structure of benzene for you. Okay, benzene structure. This is having simply carbon atoms, but at areas if it is substituted by nitrogen, this becomes a heterocyclic compound. That means other than carbon, some other element is also coming over there. So this nitrogenous base also has a structure somewhat like this. That is, it is a cyclic compound and there are nitrogen atoms present here and there. So what is that? Let us learn in detail regarding the structure of pyrimidines first. Let us learn. Pyrimidines, they are having a 6-membered ring. 6-membered ring they have. Okay. And in this 6-membered ring, let me start numbering the ring from here. 1, 2, 3, 4, 5 and 6. This is a 6-membered ring. And in this 6-membered ring, nitrogen is present in all the odd places. In all the odd numbers. Except 5. Except the 5th position. Except the 5th position, wherever odd numbers I have put, their nitrogen is there. So how to number it? You start from below and you go anti-clockwise. Okay, start from below 1, 2, 3, 4, 5, 6 like this, anti-clockwise you go. Next you look at the odd areas. Wherever I have put the odd numbers, there what do you have to do? You just put there nitrogen. So in position 1 and in position 3 I have put nitrogen except the 5th area. So there you should not add nitrogen. So this I have added now. Now what to do next? Put double strands. Double here. This is single. Again double. Single, and here also double strand will come. Alternate double and single bonds you put. Starting from here 5, 6, then alternate double and single strands. You can just put it here. So this is the structure of pyrimidine. Oh, it's very simple. Structure of pyrimidine. Now when this is getting bonded in our DNA, in order to show that bond clearly, we just remove this bond from here and instead the valency of nitrogen, you know it is 3. It is satisfied with the help of a hydrogen here. So when you draw pyrimidines, instead of this particular bond here, just take that bond here and satisfy it with the hydrogen. So this is structure of pyrimidine. Now what about purine? Let us see. Purine also has got a 6-membered ring structure like this. Okay, a 6-membered ring, and it is fused with the 5-membered ring. It is fused with the 5-membered ring. A 6-membered ring is fused with the 5-membered ring. And I have put nitrogen here and there. Isn't it? Now it's not complete. The structure is not complete. Here also, first I should go for numbering. In purines also. So here the numbering begins from here. This is 1, first position, second, third, fourth, fifth, sixth, and then 7th position, 8th position, and then 9th position. So I have numbered it. See here the numbering began from below and it is going anti-clockwise. But here in purine the numbering is beginning from here. It is going clockwise here and then anti-clockwise with the other ring. These things are small things, but these are all based on IUPAC rule and we have to follow this while writing. It's very simple for you to understand actually. Pyramid is very simple. 6 areas, a 6-membered ring is there. Start numbering from below, go anti-clockwise. Wherever there are odd positions, you put there nitrogen except position number 5. Got the structure of pyramid. Simple. Now purine, what is it? Same way, you have a 6-membered ring, but it is fused with a 5-membered ring also. So naturally there will be difference in the numbering. So here the numbering starts from here. 1, 2, 3, 4, 5, 6. One structure is all numbered and then from again top 7, 8, 9. Like this it is numbered. Here also you have to go for alternate double and single bonds. Again I am starting from here, double, single, double bonds. Okay, so here also I am putting here double bond here, single, double. Likewise it keeps on going. Okay, the bonds keeps on going like this. This is cyclic compound, it keeps on changing. So now, where to put the nitrogen? Here also you have to put the nitrogen in the odd position except the 5th position. As like here. Odd position except the 5th position. So odd position, first 1, odd position. Then 3 is an odd position. 5 I should not put. 7 is odd, 9 is also odd. I have already added there. So here also I have added nitrogen in areas where I have marked with odd numbers. Except the 5th area. There it is carbon only. There is no nitrogen. So now what I have written here, I have just drawn the structure of 2 nitrogen containing bases. That is nitrogenous bases. They are pyrimidines and the purines. Pyrimidines and the purines. Now there are many different kinds of pyrimidines and many different kinds of purines depending upon which bond is coming where. Okay. So here one more thing let me say regarding purines. See here I have removed one bond here and I have put one hydrogen here. Why did I put like this? In order to make you understand when this particular pyrimidine is getting attached to our ribose, how it is getting attached. In order to understand that I have made it like this. Similarly, here also I am going to remove this bond and instead I am adding this with the last nitrogen, 9th nitrogen, that hydrogen is added. Why this is done? Again, how this purine is going to bind with our ribose sugar. How it is getting bonded. In order to understand that, you should put the structure like this. So here it is with the first nitrogen in pyrimidine. In purine it is with the last nitrogen. So first nitrogen here with pyrimidine, last nitrogen here with purines. Simple things for you to remember actually. So compare and learn so that it will be easy for you to remember. Even if you forget one aspect, okay, this is first, okay, then purine is coming last. Like that it will come to your mind. Okay, so this is clear. Pyrimidines and purines are 2 nitrogenous bases. Now I told you there are different kinds of pyrimidines and different kinds of purines based on the substitutions. I'll give you one example. See here in purines, if I write here as 6-aminopurine. 6-aminopurine I am writing. 6-aminopurine. That means what? In the 6th position, amino group is there, NH2. This is what? 6-aminopurine. This is our adenine. We have learned ATP. We were saying all these days. ATP is what? Adenosine triphosphate. Isn't it? This is our ATP. Adenosine triphosphate. This is not adenine triphosphate. It is adenosine triphosphate. Okay. So what is the difference between adenine and adenosine? When adenine is added to a sugar molecule, it becomes adenosine. Okay. So here adenine, this particular structure, this is adenine. This adenine is added to a sugar molecule, and to this, how many phosphates are there? 3 phosphate molecules are added, triphosphate. So sugar is bonded to adenine and is bonded to 3 phosphate. This is our ATP. So why I told you only regarding the structure of adenine? Because this comes in our ATP also. So this is one purine. Which one? Adenine is a purine. And how did I make this as adenine? Because I have added in the 6th position amino group. So 6-amino purine is adenine. This way you can remember. 6-amino purine is adenine. So how to draw the structure of purine? Very simple. 6-membered ring fused with a 5-membered ring. Start numbering from one side 1, 2, 3, 4, 5, 6. And again from the top 7, 8, 9. We have numbered. It's done there. Now look at the odd positions. Put nitrogen in the odd positions except 5. And keep on putting alternate double and single bonds. When it comes to the 9th nitrogen, you remove that double bond and put there single bond H. Purine is done. Now I want to make this as adenine. How will I do that? In the 6th position you put an amino group, NH2. Enough. So you got 6-amino purine. 6-amino purine. Regarding valency and all, if you check, this double bond keeps on changing. That you know, in every heterocyclic compound, in every cyclic compound, this valencies, that double bond keeps on changing. You have learned you might be learning the properties of a cyclic compound in your organic chemistry part. So you need not worry about valency here too much. It keeps on changing. Okay. It's a dynamic molecule. So this is adenine now. Now another purine molecule is guanine. Guanine. Now how to make this guanine? It's very simple. It is 6-oxy, 2-amino. Just change the positions. The 6th position, it is oxy. I can either put here single bond OH or I can put there double bond O. Anything is okay. Okay. So 6-oxy, 2-amino, 6-oxy, 2-amino purine is guanine. So you in your this particular chapter in standard 12, you need not learn the structure of adenine, guanine and all. But you should know the structure of purines and pyrimidine. And just understand, adenine and guanine are substituted purines. That means in some areas, some other molecule is coming and substituting things. And accordingly, what is getting substituted in each position, you are getting adenine and guanine. So adenine and guanine are substituted purines. Similarly, there are substituted pyrimidines as well, isn't it? Substituted pyrimidines as well. So which are those substituted pyrimidines? There are 3 substituted pyrimidines. Okay? I am not going into detail of the structure of those substituted pyrimidines because that's not there for you in standard 12. So substituted pyrimidines are thymine, uracil, and cytosine. There are 3 substituted pyrimidines: thymine, uracil, and cytosine. So there are 2 purines which are there, adenine and guanine, and there are 3 pyrimidines: thymine, uracil, cytosine. So totally there are 5 nitrogenous bases. There are 5 nitrogenous bases in total. 2 purines. Among these 5, 2 are purines and 3 are pyrimidines. I hope this part is very clear to you. So we have completed now regarding the structure of ribose sugar. It's very simple. Then regarding nitrogenous base. What is that? 2 nitrogenous bases. Which are they? Pyrimidines and purines. Pyrimidine is a 6-member structure. Numbering from here 1, 2, 3, 4, 5, 6. All positions put nitrogen. First position you put here one hydrogen attached. Over. Structure of pyrimidine is over. Structure of purine is a 6-membered ring fused with a 5-membered ring. Start numbering from above, top, one side. 1, 2, 3, 4, 5, 6 and then 7, 8, 9. Put alternate double and single bonds. Odd positions you put nitrogen except 5. You got the structure of purines. Now here and there if you substitute, you will get substituted purines. They are adenine and guanine. Similarly, you will get substituted pyrimidines. They are thymine, uracil and cytosine. Okay, so with this we complete the structure of different nitrogenous bases. So I have told you the structure of ribose. Now we completed the structure of nitrogenous base. In the beginning itself I told you regarding phosphate. Now let's see how these three components are bonded to each other to form the nucleotide. So how are these three bonded? These three units bonded. Which are the three units? One is our pentose sugar. Then nitrogenous base is there. And another one is phosphate. So from here I think you might have understood that sugar is the core area into which the nitrogenous base and the pentose sugar are attached. So let me draw the sugar molecule first. Pentose sugar, we have seen like this, pentose sugar. Okay, here I told you it is beta pentose. We are dealing with deoxyribose. So it is HH here, second carbon. Here it is HOH, no doubt. Here it is CH2OH, one H over here. So sugar I have written. Now next I have to write, let me write nitrogenous base. Because we all know sugar plus nitrogenous base is giving you a nucleoside. So let us first form the nucleoside. Then we'll make it as a nucleotide. Okay. So nucleoside. So for nucleoside, sugar I have written. Now I have to draw. I have to attach it to the nitrogenous base. So let me first take the simple nitrogenous base pyrimidine, a 6-membered ring. 6-membered ring. Okay. Alternate double and single bonds. And in odd positions there is nitrogen. Nitrogen position number 1, 2 and 3rd position. Okay, this is position number 1. And here I have attached this to a hydrogen molecule here. So this is how the structure of pyrimidine is. Okay, structure of pyrimidine. Now what I have to do? I have written a nitrogenous base over there. I have written a sugar. This is my sugar. This is the nitrogenous base. Now I have to link these two. This linkage I have to show. Where is that linkage is the question. The linkage is like this here. The H here and the OH here like this. One water molecule will be removed. H2O. One water molecule is removed from here. So what is left? This H here is directly bonded with this N here. This H there is directly bonded with that N. Isn't it? So I can now write the structure here like this. So what did I get here? So here the bond is here like this, and this is bonded to the nitrogen of the nitrogenous base. Is bonded to the nitrogen of the nitrogenous base. Like this. Is bonded here. So rest of the areas it is same, that there is no change. There is no change in rest of the areas. The only change is that here, this here is directly bonded to the nitrogen. Why? Because one water molecule is removed. So in order to show this, in order to make you understand this only in the beginning here, I have, instead of double bond here, I have taken the bond here and I have put there single bond H. Only then you will understand how this is getting formed. Okay. So what I have written here is a nucleoside. Now how to make it as a nucleotide? I have to add phosphate. Add phosphate to this particular structure. We are going to get a nucleotide. Isn't it? Nucleotide. Now phosphate, as in the beginning I told you, It is having a structure like this. This is the structure of phosphate. Here also, if phosphate has to bond it to the sugar molecule. So phosphate is getting bonded to the 5th carbon, the 5th position. Where is the nitrogenous base getting bonded? It is bonded to the 1st carbon. And phosphate is bonded to the 5th carbon, first and last. To the first one, nitrogenous base comes and attaches. It becomes a nucleoside. Now phosphate gets attached to the last carbon. Then it becomes a nucleotide. So here, how it is getting attached? Again, water molecule is getting removed. H2O. 2 hydrogen and 1 oxygen is getting removed. So now, how is the bond? So here, H2O is gone. H2O is no more here. It is gone. H2O is not there. So I can extend this oxygen. I can extend this oxygen towards the center, little bit center like this, and I can show the bonding. Now see, phosphate is also bonded to sugar molecule. And this bond is actually an ester bond. Ester bond. Phosphoester bond. Phosphate is there, oxygen is there in between. This is a phosphoester bond. So now to the sugar, I had added nitrogenous base. I also added the phosphate molecule. So what I got here? I got a nucleotide. I got a nucleotide. Now I told you n number of nucleotides are added one after the other to get the nucleoside. So our next attention is that how this one nucleotide is attached to the next nucleotide. That is, see, our interest next one is here. We have a nucleotide, sugar, bonded to any nitrogenous base. Here it is bonded to phosphate. Again, another nitrogenous base, nucleotide I am drawing here, sugar, bonded to the nitrogenous base 2. This is nitrogenous base 1. And again phosphate. So always in this structure, what is changing is only the nitrogenous base. It can be adenine, thymine, guanine or cytosine. Any nitrogenous base can come over there. But sugar and phosphate remains the same. Okay, that's why I have written here nitrogenous base 1, nitrogenous base 2. Instead of this, I can also write adenine, thymine, guanine, cytosine. Anywhere I can write. Anything I can write like there. So here, see, this is one nucleotide. This one. This is another nucleotide. In order to make it as a nucleic acid, I should draw a bond between these two. And how are they bonded? The bond is again between the sugar of the first one with the phosphate of the second one. This is the bond. The sugar of the first one is bonded to the phosphate of the next one. Again, if I draw a third one, a third nucleotide, if I am drawing sugar here, a nitrogenous base, third nitrogenous base is coming here, another phosphate is here. So this is a nucleotide. Now how to join it with the second nucleotide? Link the sugar with the phosphate. This is the third nucleotide. So again, now fourth one, if I am taking sugar, nitrogenous base 4 linked to phosphate. Again, if I have to link the third one with the fourth one, there is no doubt, take the sugar of the previous one, link it with the phosphate of the next one. So fourth one I have linked. So I have added here 1, 2, 3, 4. All these bases I have added. All these nucleotides I have added. So ultimately what I am getting here? I am getting here a nucleic acid. So when nucleotides are bonded one after the other, I am going to get a nucleic acid. So here what is left to explain is this particular area. How is this bond formed? Already I told you how this bond is formed between sugar and base and how this bond is formed between sugar and phosphate. It is all removal of hydrogen and then adding up of the bonds over there. Now our next question is, how about this one? How the sugar of the previous one is bonded to the phosphate of the next one? That is our question. So let me explain to you that by drawing the structures properly. by drawing the structures properly. Okay, so here again I have to draw that. I am taking the first one here. I am drawing the sugar. Sugar molecule is there. This is bonded to nitrogenous base. So that nitrogenous base, if I am taking it as a pyrimidine, it is N here, it is bonded to this N here. Okay. I'll also show you if it is purine, how it is, that I'll show it to you. Okay. So this is bonded here. Second position, it is HH, only then it becomes deoxy. Third position, it is HOH. Fifth position, it is, fifth position, it is CH2O, and that O here is bonded to our phosphate. Okay, rest of the valencies is same. This is the structure. There were 3 OH groups here. Now 1 OH got bonded here. This is my first area. This much I have drawn there. Now let me draw the same way, the second one, to show the second one. Second one, I am putting it here. Sugar molecule. There is no change in the sugar molecule. This is bonded to nitrogenous base. So now I'll show you. In the second one, let me tell you, it is bonded to the purine. Purine means a 6-membered ring. It is fused towards a 5-membered ring. It is fused with a 5-membered ring. Okay. There are double bonds. Single, double, single, alternate, double and single bonds are there. And there are nitrogen in the odd position. This is position number 1, 2, 3, 4, 5, except 5. Then this is 6th one. Then again the 7th position it is there. 8th and 9th position it is there. So here you remember I had drawn here NH, the last position for purines. Okay, so that H is actually replaced here. Like in this case H was replaced and this is bonded to the last nitrogen. So in the case of pyrimidines, sugar is bonded with the first nitrogen, first carbon, first nitrogen. But in the case of purines, first carbon sugar, that is bonded to the last nitrogen, the 9th nitrogen. That is the difference. So these positions, small things, though they are, it is very important. Okay. Exam point of view, it is very important. So here it is bonded to the last nitrogen, 9th nitrogen, it is bonded. Rest of the things are same. HH, HOH, CH2. CH2 is bonded to O and that O is bonded to P. Apart from this, it is having the usual H and OH areas. Okay, so what I have drawn is this particular area, second sugar. Sugar, second nitrogenous base and phosphate. So this was sugar, first nitrogenous base phosphate. This is sugar, second nitrogenous base I have drawn and phosphate I have drawn. Now what is the question? This is the question. Or this is the question. How to link the first one with the second one? How to link the first one with the second one? This is the question. So how to link it? Where is the linkage between the sugar of the first one and phosphate of the next one? Sugar of first one, phosphate of next one, isn't it? So here I can simply draw like this. H2O. H2O. Here also, what is lost? Water molecule is lost. H2O. So what is left here? This H will be bonded to this particular O. So here, where is the bonding actually? It is with respect to the third carbon. Third carbon of the sugar is bonded to the phosphate of the next nucleotide. Now again, if I draw here another sugar molecule with phosphate, okay, double bond O, single bond OH like this, Okay, if it is bonded like this, here of course there will be a nitrogenous base. Where the bonding will be? Sugar is bonded to phosphate. Always it is like that. Sugar bonded to phosphate. Sugar bonded to phosphate. Sugar to phosphate. Like that it is going. So where is the bonding? Which part of the sugar is getting bonded? It is the third part. This region is getting bonded to the phosphate here. So again water molecule is removed. H2O. What is left? H is bonded to this particular O. Let me show you that bonding very clearly over there. So here in this area, H2O is gone. H2O is gone. The third position. H2O is gone. And what is left here? This oxygen. This oxygen is actually bonded. This oxygen, I am putting it here for you to understand. Okay. So you can look here, see children. Phosphate is bonded with one oxygen towards the sugar. And phosphate is also bonded with an oxygen with the previous sugar. So phosphate is actually involved here in two ester bond formation. Okay, one is here and another one is here. It is actually forming two ester bonds over there. Two ester bonds. So you call this as a phosphodiester bond. Phosphodiester bond. Two ester bonds are there. So that is actually this bond. The name of this bond is phosphodiester bond, or it is the phosphodiester bond which links one nucleotide with the other nucleotide. So two nucleotides are linked with the help of a bond called as phosphodiester bond. So the bond that is running throughout the nucleotide is phosphodiester bond. Like for amino acids are linked by peptide bonds, nucleotides are linked by phosphodiester bond to form the nucleic acid. So this is just one strand of the nucleic acid that I have shown you. So I have shown you just one strand of the nucleic acid. Okay, likewise our DNA is having one more strand. So what is the other strand? How that is formed and how this is linked to this particular strand? Okay, which is the bond between them? How is that bond formed? Is it a weak or a strong bond? What is the significance of that bond in the formation of our genetic material? All these will be dealt with in the next video. Okay, so in order to understand all these things, I think you will have to subscribe this channel. If you subscribe the channel, you go to the playlist, click on the videos one after the other, okay, and do watch those videos. Come with your doubts. Feel free to ask the doubts. In the previous chapter also, if you have any doubts, feel free to ask. I'm very happy that so many comments are coming. You are asking doubts and I'm able to clear it also with you. Okay, so keep doing it. Keep doing it. Stay home. You all stay healthy. Okay, spend nice time with your relatives, with your friends, and have fun. At the same time, spend time for learning as well. What we have completed is a very important aspect of our genetic material. In this chapter, we have gone down towards the molecular basis of our inheritance. Having completed the principles, now we are dealing with the molecules, and the first and the most important molecule is our DNA, and its general structure only I told you. Till now the structure of DNA is not complete, so for that I'll come in the next video. Thank you for now.